Okay, so welcome to our final exam review for Bio 205. First thing I want to say is y'all have done a fantastic job with getting this far. Um, I've been very happy and glad to be part of this class. Y'all been very good, and I really hope that this is a good experience for you all, especially as a biology class, but also some of you might be introducing or being introduced into like research, which is fairly unique to Phoenix College, that we do kind of research things that usually are reserved for like the big four universities like ASU and NAU. So at the most part, I hope you take advantage of the opportunities that you got in this class, and hopefully you can carry that on wherever you go after this. So for 205 review, I'm going to start with the Unit 4, just because that's the most recent and we haven't been tested on that at all. And then I'll go back and cover Units 1 through 3, and then I'll leave it open for any questions. If we do run out of time and there's still a lot of review left, I might close this and reopen another one just for questions. There you go. Okay, so just some logistics for the exam. Just know the exam will be during your normal lecture time, either Monday or Wednesday, starting at 8:30 a.m. in DB123 down in the lab building. You'll have 2 hours to take the exam, but if you finish early, you'll finish early. You do have to use the Respondus Lockdown browser. We do have laptops available. However, if you want to bring your own device, I would recommend just bringing it if that's what you're comfortable with, if that's what you've taken your exams on. Why change anything? Just bring your device, get on the internet, and we'll take the exam together. There is going to be a passcode, so you can't take this exam from home, logging at the same time. Dr. Cotter has the passcode. She'll give it to you during the lecture in the room, and then you'll put that passcode in to let you get into the exam. Overall, there's going to be 84 questions, multiple choice and short answer. And by short answer, we mean something like you might be given an image and it might be stained purple, and it's asked, hey, what type of bacteria do you think this is with a purple stain? And you'll just type in gram positive and then next, and you move on. It's nothing too difficult. It's all stuff you've seen before. You can kind of see that Dr. Cotter tried to ask you similar questions during our last lecture, kind of going back to stuff from Unit 1, 2, and 3. But for the most part, short answer, 1 or 2 words, maybe Koch's postulates, something like that. Excuse me. All right, starting on Unit 4. So this is the unit that we've just finished up. One thing to mention, the exam is broken down into fourths. So you're going to have 25% on Unit 1, 25% on Unit 2, 25% Unit 3, 25% of Unit 4. So it's fairly evenly spaced. For Unit 4, it was only really one chapter, which was pretty nice, Chapter 13. The way these questions are going to work is they're going to be mostly revolving around the infection chain. And so if you remember what this was revolving around, we talk about what is the illness, what's the causative agent, which is always the microbe, right? So you should always have a microbe for causative agent, not a nail, not people, not droplet, not food, no strep throat, whatever. It should always be a microbe for causative agent. I'll reemphasize it because people do miss this. They'll call the strep throat the causative agent, for example. Then we're going to go into reservoir. So where is the microbe hiding? Where is it living? Where is it residing, etc.? Things like contaminated food, stagnant water, dirt, soil, another person like through direct contact, or another living organism like a dog, for example. And then the mode of transmission, how did the microbe get into the host, like me, from another reservoir, right? So a vehicle, I like to think of it as like you're traveling, right? So you travel through air, land, and sea. So air is through the air, right? Sea through water, and then driving, that's kind of one thing I don't really have for, but I think food. So food, air, and water, that's your vehicle transmission. Vector transmission is, or transmission, that's done through bugs, things that don't give you the illness directly, but they are carriers of the illness. So think of fleas, mosquitoes, ticks. The mosquito isn't giving you malaria, it's transmitting malaria from one place to another, and it just gives it to you through its own mechanism. In direct contact, that's usually stuff that happens with non-living things, right? So if I touch a contaminated knife, or if I get cut by a contaminated knife or a nail, that's your indirect contact. Direct contact is pretty straightforward. The one thing that I would tell you about direct contact for some of the other ones is the other thing that's direct contact should be something that's living and is ill or infected. So if my wife was sick with something and I kissed her, that would be direct contact because she is infected, transmitting something into me. A nail, soil, water, those aren't infected. They just happen to have that microbe with them. If you have any questions on how this works, just feel free to ping me or message me. But this is kind of where we start getting into like semantics of, the mosquito gave it to me, but the mosquito wasn't really infected. Mosquito just carried it from one place to another, that kind of thing. But we did go over some examples on page 1 of the Unit 4 study guide. As long as you have that filled out and you can group those all into their appropriate columns, like the illness, causative agent, reservoir, mode of transmission, you'll do fairly well on the Unit 4 portion. Some of the other things that you might have brought up is the stages of illness. So remember that we have our inoculation period. This is where you initially get sick, but you may not show any symptoms. So this is where the microbe, the virus starts spreading. It starts damaging your cells, starts hiding. Usually for most illnesses, you might have a few days to like a week of an inoculation period where you don't even know you're sick, but the invasion or the illness has already started. Prodromal period is where you start feeling the mild symptoms. That's where you start feeling like, I'm a little weak today, or a little tired, or a little sore. Maybe I have a little light cough. Yeah, we've all been there where we kind of know when we're getting sick. That's prodromal. The getting sick portion is the prodromal. You've already been inoculated. The illness has already been in your body for a couple of days to a week. Now your body's just starting to show the signs of those illness. The active illness portion, or step 3, is the most severe. This is where you have the fever, the chills, the diarrhea, whatever. This is where you're on medication, sitting in your bed, lying down, sleeping, or have to go to the hospital. That's the active illness portion. This is usually the make or break point for most people. And what I mean by that is this is where either the illness is going to win and your body's not going to be able to fight it off. Medical intervention is not going to be able to do anything. And then unfortunately, you move on to either pass away or have some long-term effects. Or this is the part where the illness gets to its peak before your body and medical intervention bring it back down to take control of it. That portion we call the decline period. So now the immune response is strong enough to deal with the infection and then some to actually start killing it. Dr. Cotter has talked about how our immune system can defend itself against basically everything. The most of the time that we have issues is that the immune system needs time, and sometimes the illness or the infection does not give you the time to catch up. And so as long as your immune system can deal with the infection, either by itself or with using a booster shot or medication or medical intervention, for the most part, we all go through the decline phase. Obviously, the worst case scenario is that your body cannot handle the illness and it causes too much damage to where your body can't. And then the last stage is the convalescence period. So now this is where you're, okay, fever's gone, chills are gone, I'm no longer vomiting, I'm no longer throwing up. I just feel lightly weak, but now I can start actually feeling normal or like myself again. And usually the convalescence period could last a few days. For some illnesses, especially depending on how much damage is caused, this could go on for months or weeks, months, or even sometimes years. Just kind of a side note, if you've ever heard of the term long COVID, that's something that they've been looking into as people still have signs of illness, like months or years after having COVID. And because the virus is so new, we're not really sure what that could mean for you in 10, 20, 30 years. So just so you've heard of that portion, long COVID, that's kind of happening in the convalescence period where your body should be recovering, but for some reason you still have some signs that you were ill months or years before. And then just kind of quickly go over the endotoxins versus the exotoxins. Primarily, endotoxins are in gram-negative bacteria. They're typically lipids or fats. They are membrane bound, so they are attached to the bacterial wall. They're typically heat stable like butter. Butter can melt, butter can be sat out on the countertop. If you just leave butter out, it doesn't melt, doesn't change, it just stays as its block or whatever tube is in there. And if you add heat, the butter melts and looks different, but it's still butter. It still tastes like butter. It doesn't actually change much about the butter. Lipids or the endotoxins create systemic effects. So because they're attached to the bacteria, they kind of travel with the bacteria. And depending on where that bacteria ends up, your body will have a generic effect or a generic response to it, like the fever, inflammation, that kind of thing. And some examples of that are the LPS or lipopolysaccharides. For exotoxins, we're talking about stuff that is primarily secreted or released by invading bacteria. Primarily, these are gram-positive. They are proteins in nature. Like I mentioned, they're secreted, and they're not necessarily heat stable. So proteins are not heat stable. You change the temperature of a protein, it's going to change its form or the way or its shape, which then changes its function or how it can operate. For the most part, because these are secreted and they're protein based, they look for specific cells to target. So a protein that affects a liver cell won't necessarily affect a lung cell, for example. Some examples of these are like the neurotoxins or the super antigens released by Staph aureus. And let me look at the chat real quick because I've got something in there. Do we have to know agents for that? What do you mean by that? Like the bacteria, you mean? I would, give me 1 second. I would know not so much for the endotoxins, but if you look at page 4 of your study guide, there's a list of exotoxins, the enterotoxins, neurotoxins, cytotoxins, the humanologic and super antigens, and we have examples of those. I would at least have 1 or 2 examples for each of those for page 4 of the study guide. To answer your question, and if you need that page, anyone in the chat or just the group, just let me know and I will send you my completed Unit 4 study guide so you at least have that to study. That's a good question. So you don't necessarily need to know the agents for endotoxins, but exotoxins, because it is part of the study guide, we do ask you for that.
Hello, I need it.
Okay, perfect. I'll send it. It sounds like everyone in this group so far has chimed in. I'll just send it out to all the students in an email. Thank you for bringing that up. Excuse me.
So you will send me by email?
Yep, I'll send it through your Canvas email.
Okay.
All right.
So you say handout page 1 or page 2 when you started the live?
Sorry, what's that, Sarah?
The handout, which page?
The unit study guide, I'll pull it up. So the Unit 4 study guide, and it's page 4 has your list of it, but I'll send out my completed study guide so you have all the pages.
Okay, Unit 4 study guide. Okay, I got it.
Yep.
So we have to bring it to the class Monday?
Yes. Dr. Clotter does ask that you bring the Unit 4 study guide for class, and I think if it's completed, she'll give extra credit. I'm not 100% sure on that because I won't be there, but I think that's her intent of doing it.
Okay.
But I'll send you mine. You guys can fill it out, and then you should have it all completed for Monday or Wednesday.
Okay, just send me an email, please.
Yep, definitely will do. Thank you, sir.
Oh,
Okay. This is the list I was talking about. So a little bit forward. So these are the types of exotoxins, and you can see how I have some examples of like Clostridium tetani, which is part of our chapter 13 study guide. Streptococcus pyogenes, we talked about that a lot. Staph aureus, etc. But again, this is in my Unit 4 study guide, and I'll send that out to you so you can have a good copy of that. And then the last portion that we talked about was or other things to talk about are the virulence factors. So what makes an infection or a disease more deadly or more contagious? So we look at things like the pili, the viral spikes. We also look at stuff that they use to hide from our immune system, like the glycocalyx, also antibody receptors and enzymes. And also we look at how much damage the bacteria could cause in the body. So you have some bacteria that could come in and just do like damage to the respiratory system. I'll use the flu or influenza, for example. But then you have a virus like COVID-19, which uses a very specific receptor. I believe it was, I don't recall the name of the receptor, but the receptor itself is systemic. So a lot of our body systems use this receptor. And so it can attach to things like respiratory cells and liver cells and cardiac cells and a bunch of other stuff. So things like that make it more deadly versus something that only attaches to liver cells or respiratory cells, for example. And then just a quick reminder of the definitions. So we have 4 things we really went over: endemic, outbreaks, epidemics, and pandemics. So something that's endemic is something that occurs in a local population at a very constant rate. Things like valley fever in Arizona and California. We have a very stable rate of valley fever in this area. So valley fever is considered an endemic. An outbreak is something to say that it just suddenly is something that's much higher than it normally is. So think of like an E. coli infection. So we have bad meat. Bad meat gets eaten in Arizona. We have an outbreak of E. coli in the local population. It's very quick. It's sudden. And typically those stay as outbreaks. They get resolved. They figure out where the meat came from. They throw the meat away at the grocery store. People recover and then move on. An epidemic is something that is an outbreak, but it lasts a lot longer. So something like an E. coli outbreak from food, it probably won't last longer than maybe a few days or a few weeks, whereas an epidemic will last for much longer, talking the months or years. And then a pandemic is usually an epidemic that's happening on different continents at the exact same time. So things like the flu, COVID, plague, etc. Okay, so that's basically it for Unit 4. Like I said, there's not much to it. There's only 1 chapter, but I'll send out my study guide. You guys can look that over and prepare for the exam. All right, so going back over Unit 1, here's some of the things that people tend to miss in my experience here in the last 4 years. Folks' postulates, again, fairly easy, but people tend to miss this one. Again, remember the four steps of Koch's postulates. We're going to observe the patient for signs of illness and isolate the microbe. Remember that signs are things that can be seen by the doctor or by a physician, and the symptoms are what the patient's complaining about: the nausea, the upset stomach, the headaches. We can't measure headaches, that's a symptom, but we can measure things like fever or temperature or blood pressure. We can see open wounds, that kind of thing. Next, we're going to culture and identify the microbe in the lab using different tests. These are where your biochemical tests come in. You've done a lot of these throughout the semester. Remember your microscopic, macroscopic, biochemical tests, DNA analysis, serological tests. These are some examples. Step 3 is to infect a new host, either different types of cells or something that's living. and observe the same signs of illness. And then in step 4, you're going to make sure it's the exact same microbe by repeating the same steps you did in step. Very quickly, remember that we have different types of cell parts. Again, kind of like in Unit 1, while we do care about cell parts, we really care about you being able to use cell parts to identify an organism. So if we say things like GP120 viral spike, that should tell you, okay, that's a virus, it's not a fungal cell. We talk about things like a cell wall versus a cell membrane, talking about pilus, gram staining, purple versus pink. The things that we've been using this whole semester to differentiate different types of microbes, that's what you really care about in the cell parts. I would definitely go back and review units 1, 2, and 3 study guides so that you can remember some of this stuff. The exam is all going to be based off the 4 study guides that you already have. So parts, again, kind of just going back over that. I'm not going to spend too much time on this one. Just look at your notes. Again, we also use bacterial shapes and sizes to differentiate what types of bacteria that we have. So things like the Staphylococcus or the Streptococcus, the flagella type. If you have a flagella question, you might have one. So if you don't look at it, I think you'll probably be fine. But the things that we're pretty much been doing the whole semester, the shapes, rods versus circles, that kind of thing, I'll definitely. And then just kind of go back to my rant from earlier. You got to be able to identify the microbe based on what things that we give you, either cell shape, cell size, color, cell parts, things like that. Viral infection and replication. Remember, these are your 6 steps. Remember, attachment, entry, uncoding, replication, assembly, and release. And specifically, when we talk about this, you want to be specific on the types of viruses that we're talking about. We've only dealt with HIV and influenza. We're only going to have HIV or influenza on the exam. Don't worry about studying anything else. Just remember, HIV uses the GP120 and GP41 viral spikes. They infect CD4 plus helper T cells, and they utilize reverse transcriptase, which means that the HIV RNA gets backwards written into DNA, and the DNA gets written into viral RNA. Versus things like influenza, which use hemagglutinin and neuraminidase viral spikes. They infect the respiratory epithelial cells, and they use RNA polymerase, which means the RNA goes into our cell, and then it just starts making proteins from the RNA itself. That's unit 1. Excuse me. Unit 2. Very quickly going over different types of conditions. Remember that we have different environments determine the type of microbes that grow in there. So things like temperature, we'll have things like saccharophiles, mesophiles, thermophiles, hypothermophiles. For oxygen, we have the strict or the obligate anaerobes. We have strict or obligate anaerobes. We have facultative, which like either low oxygen or high oxygen environments. We have the aerotolerance, and we have the microaerophiles. And then pH and salt, again, we have the acetophiles, neutrophiles, and alkalinophiles that like different types of pHs. So any pH below 7 is acidic, a pH of above 7 is basic, and a pH equal to 7 is a neutral pH. And then salt, we have the halophiles and halotolerant. I don't think we covered this much besides the definition, so I don't really expect a question on this, but it's there if you need it. Bacterial genetics, so remember we have horizontal gene transfer, which is basically saying how bacteria can share information with each other without actually replicating. So the 3 big ones that we talked about for this class are conjugation, transformation, and transduction. Conjugation is, think of it as bacterial sex. So you have the sex pilus, two bacteria come together, they share genetic material, and the one of the bacteria typically ends up getting some type of antibiotic resistance from the other bacteria. And now you have 2 bacteria that have antibiotic resistance. Transformation is the use of some bacteria are able to eat other parts or destroy other bacteria and eat their pieces. And by eating those pieces, they're able to absorb some of the DNA or the antibiotic resistance from the other bacteria, and then they build it into themselves, and then they make brand new cells from that. And transduction, so if you're in the Wednesday class, you know what this was all about. We used the phages, where a bacteriophage, when it's trying to reproduce, it accidentally grabs a piece of DNA from a bacteria, moves it to another bacteria. Now that other bacteria gains antibiotic resistance accidentally from that bacteriophage. So the virus is used as like a vehicle to transfer a piece of DNA from one bacteria to another. Energetic shift for energetic drift. So drift, remember, is a small change in the viral spike structure, typically resulting in point mutations. And the shift is a major change in viral spike structure. It changes very quickly. And it's a result of genetic recombination where multiple different viruses come together in a similar host or one host, and then they can divide and become these brand new types of viruses that our bodies have never seen before. I did get a 10-minute warning, so I'll try to be a little quickly on this one. But some of these are very similar to my other exam reviews. So if you want to go back and look at my Unit 1, Unit 2, Unit 3 reviews, I'll cover some of this stuff as well. Methods of control, just remember that we have different types of control methods. We have physical and chemical, and we always try to look at ways to sterilize, disinfect, and be something that's antiseptic. So sterilizing is killing at 100%, disinfecting, killing at less than 100%, and antisepsis is for killing less than 100% on living surfaces. These are some examples you see here, but again, go back to your Unit 2 study guide, page 7. That way you can have those memorized in your notes. I really just focus on the big three, the O's, the aldehydes, halogens, hydrogen peroxide. Know some sterile techniques, some physical sterile techniques, some chemical sterile techniques. I don't think you have to go over all of them. If you want to, it's good to know, but at least know your big ones, your sterilizations for chemicals, sterilizations for physical methods. And I think if you know that, you should be okay. And then the antibiotics, we have different types of antibiotics that attack different portions of a bacteria or a virus. So here are just some examples that we have in our notes. Again, go back to page 10 and review the antibiotics for this. Again, looking at more of examples of what kind of bacteria or what kind of antibiotics target like the peptidoglycan cell wall or the waxy cell wall or DNA, et cetera. And then Unit 3, we just did this, but remember that we have different types of barriers. So our first line of defense, we have physical and chemical barriers. For the most part, things like our mucous membranes, our mucus, our cilia, or our hairs, those are found in multiple different places like the respiratory, GI tract, and reproductive system. Chemical barriers, we're talking about mucus, saliva, the lysozymes, lactoferrins. Having a picture like this where you have it broken down to what specifically is in each organ system is very useful. But the big ones we'll probably talk about are respiratory, GI tract, or reproductive. Resident microbe or resident flora, those are just the things that live inside our bodies that compete with resources with foreign invaders. And that way there's less resources for the infection, the Staph aureus infection, to take a hold of it. Our normal flora or our resident flora or resident microbes, they're there to block attachment. They compete for resources, and they also secrete their own antibiotics, which helps attack other foreign microbes. There we go. Second line of defense. This page is awesome. Just remember that in every second line defense, you have neutrophils and macrophages, and that the macrophages are always the cell, the antigen presenting cell that always goes to our generals or CD4 helper T cells to then tell them to start the specific or third line response. This is from your notes. It's also on, I think it's page 14 or page 24 of your handout. But just being able to remember that these are the second line cells and what they do. But our big ones, neutrophils and macrophages, no matter what the illness is, you should always see these two. And then third line defense, we try to differentiate between whether they are an intracellular or inside the cell response, or an outside the cell, extracellular or humoral response. The only difference between the two is that for the intracellular response, you're looking at the Th1 helper T, CD4 helper T cell, because you're number 1. Outside the cell, you're priority number 2, and so that's Th2. Remember that Th1 leads to the cytotoxic T cells and memory T cells, and they use perforins and granzymes to go around, pop open your infected cells, and then destroy the bacteria or the virus, in this case, the virus. For outside the cell, that is everything except for a virus. So intracellular, virus, Th1, extracellular, that's bacteria, fungi, parasite, allergen. That is a Th2 response, releases plasma B cells, memory B cells, and then the memory B cells or the plasma B cells go around your body. They release antibodies, and there's 5 different antibodies. I recommend knowing the 5 types, at least the big 4, the IgM, IgG, IgA, and IgE. And just know that antibodies do not kill. They just agglutinate, neutralize, optimize, and perform complement fixation. Agglutinate means they clump together, so they make these large globs that allow the macrophages to then come and just eat them all up. Neutralize, they attach to where the bacteria would attach itself to like our cell walls. So instead of the bacteria being able to attach to like my throat, the antibodies circle the bacteria and it prevents the bacteria from actually attaching to anything. Optimize is like a seasoning. So the antibodies season the bacteria so that our macrophages, when they're out doing whatever, they can smell or detect that, hey, this new bacteria has been seasoned. It tastes like carne asada. It's going to come over and eat it all up. It just prioritizes the macrophages to eat that over something else. And complement fixation, that involves complement proteins. It just tells the complement proteins, hey, attach here so we can go ahead and destroy this bacteria. Okay, and then final tips for this. Again, everything on the exam will be covered in your Unit 1 through Unit 4 study guides. So I'll just go back and review them. Anything that in there is fair game, but anything outside of that, I wouldn't necessarily worry about. It is multiple choice for most of it, and then you do have the short answer questions. Just remember that if you're not sure on a question, get rid of the ones you don't know, and then give yourself a better chance of guessing the right answer. You can move ahead on this one. So if you're stuck on a question, don't sit on it for 5 minutes. It's, you have 2 hours, so that's 120 minutes, and you have 84 questions. At most, you should spend 1 minute, maybe like 1 minute and 10 seconds on a question. If you get to that question and you do not know the answer and don't feel like you're coming to the right answer, just move on to the next one. And a lot of times, some of the questions later on might help you remember what that question was. The last thing I want you to do is to run out of time and not finish the exam because you spent too much time looking at Unit 4 questions when you, like in your head, could have probably done better on Unit 1, 2, and 3, and then you go back to Unit 4. This doesn't really apply for the quiet place. We're going to be in the DB123 lecture room, so just meet in there, be on time. Dr. Clotter is not going to give extra time, so if you show up at 9 o'clock, you have from 9 to 11:30, or sorry, 10:30, so you're losing time. So show up there, be ready at 8:30, so you have the full 2 hours to take the exam. And then lastly, get some good night's sleep, eat something before you show up, and bring a coffee or drink to the exam. That was the Rose. That was the so aldehydes, halogens, and hydrogen peroxide. So those are the big 3. Those are the only 3 chemicals that will sterilize. So I would definitely look at that first. Um, do I know of any good practice quizzes? I do not. I can send some stuff out. I think I'm going to get kicked out of Zoom here in like 10 seconds. So I'll send some things out here. I'll see what I can find, Christian, and send that out to you.